In the last video, we talked about how our ears are used for hearing. Now let's discuss the other function that our ears are responsible for, balance or equilibrium. First, let's discuss a few disorders of the ear. So otitis media is the technical name for an ear infection, which is when fluid fills that middle tympanic cavity, our middle ear. And children, again, I said, are most prone to this because of their horizontal tube. And so the fluid doesn't just drain back into their sinuses or their nasal passageway, it'll instead fill that tympanic cavity. And so what people will do is if you have chronic ear infections, they recommend that you get PE tubes or pressure equalizer tubes, sometimes called tympanostomy tubes or even eustachian tubes because the tube itself is called a eustachian tube, and so they put eustachian tubes in the eustachian tube. It's a little confusing, but you can call them PE tubes, and this is recommended for children with chronic ear infections. It holds that tube open so that the fluid can drain. Deafness is any degree of hearing loss, and there are two main kinds of deafness. Conduction deafness, where there's an issue transmitting the wave through that external or middle ear. An example would be if you had a buildup of earwax and all of a sudden you couldn't hear very well. That would be conduction deafness. Sensory neural deafness is when there's a damage to the nervous system structures. So damaging the hair cells, damaging the cranial nerve, or damaging the auditory cortex. And this is a lot harder to fix. Hearing aids will help you amplify sound waves, which are good if you have damage to just a few hair cells. This is usually what older folks get. Or if you have damage to the auditory pathway, you might need a cochlear implant, which will bypass the damaged part of the ear and will stimulate the auditory nerve directly. And this is best for severe hearing loss. So if you think about an 86-year-old man, he has suspected hearing loss. He has a hard time following conversations in loud environments. You want to think about what hearing loss he's experiencing and what frequency would be hardest for him. So pause the video and think about this scenario for a minute. Okay, this is called presbycusis, which is age-related hearing loss. And this is generally sensory neural in nature because it's damage to the hair cells that occurs with time. And usually the damage occurs first to our high-frequency noises. And so older folks will generally have a hard time hearing high-pitched noises first. This is also the noises that you will start to have a hard time hearing if you also damage your ears, say from explosions or loud music. If you play your headphones really, really loud, you're going to damage the hair cells, starting with high frequency hair cells. And like all neurons, you cannot replace them. So once you have damaged your hair cells, they are gone for good. So please make sure your AirPods not too loud. Okay, so we talked about the different parts of the ear. Make sure you can think about what each part does and how sound waves flow from the outer ear into the middle ear and the inner ear. Okay. So let's talk about equilibrium now. Let's talk about equilibrium. So the vestibule, this middle region, it houses static equilibrium. And static equilibrium tells us where our head is in the up-down position. The receptors are called maculae. And what's interesting about static equilibrium is that it's not a fluid that will move over the hair cells here, but instead it's something called otoliths, which are little salt crystals. So you have these little salt crystals, and they'll flow and they'll bend the hair cells, letting you know that you're moving your head down or up. And so you can see this picture here again. As you move, the otoliths will move and they'll bend the hair cells. We also have a dynamic equilibrium, which is housed in our semicircular canals, with each semicircular canal corresponding to a plane of motion, either the X plane, the Y plane, or the Z plane. And the receptors, the hair cells here, are going to be in an area called the ampulla, which has a structure called the crista ampullaris region, and it has these little cupula, which are the hair cells here. And this is also going to be fluid moving. So we're going to have endolymph fluid, which is going to move in the ampulla, and then in the cupula specifically, it's going to bend those hair cells. So again, we have our little cupula hair cells. The endolymph is going to move as you spin around in circles. Okay, so take a minute and see if you can compare static and dynamic equilibrium. Finally, let's quickly talk about smell and taste. So we want to describe the location, structure, and function of our olfactory smell and our taste receptors, and name our 5 taste sensations. So our olfactory receptors are on the roof of your nasal cavity, which is why sniffing is so important, because sniffing is going to flow more air over those receptors. And these are chemoreceptors. So what they're detecting is a chemical in these olfactory hairs, these cilia here, and they're going to send that information through the olfactory receptor cell. The axons will form the olfactory nerve, and it will send the information first to our limbic system and then to our cortex. And the limbic system is important because the limbic system has to do in part with memory, and this is why smells and memories are so linked. So if you think back to your grandma's house, for example, you might have instantly a smell that comes to mind. Or if you go into your friend's house, if you've been there for dozens and dozens of times, it'll have a distinct smell. It'll smell like their house. And that's because we associate really strongly smells with memories. And you can use this as a student to your advantage. So if you have a specific scent that you wear while you study, if you wear that scent while you take an exam, it might help trigger those memories. So you can use peppermint oil, for example. Taste buds are mainly on the sides of our tongues, or on the sides of these villi papilla, which are on the tops of the fujiform papilla, and that's where our taste buds are located. And they're going to send the information through a few different cranial nerves, 7, 9, and 10. And the hairs are located through these pores. And your tongue taste buds are actually replaced really quickly, which is a good news. So if you ever burn your tongue, you might not be able to taste for like a day, but usually by the next day you can replace it. You can taste again. And that's because these top epithelial cells replace themselves very, very quickly. And so they can be easily damaged and they're fine. And that way they protect the sensory nerve that's deep in your tongue. We have 5 taste sensations, and they correspond to different chemoreceptors. So again, smell and taste both use chemoreceptors, which detect chemicals. And so bitter is going to detect basic chemicals, mainly in the back of your tongue. Sweet is going to detect sugars and certain amino acids. Interestingly, sweet also detects lead, which is why kids like to eat lead paint, because apparently it tastes sweet. So make sure you hide the lead paint or remove the lead paint. Sour is going to be for things that are acids. Salt, you're going to pick up on those ions, so the sodium, the chloride, the potassium, that's going to taste salty. And then we have a 5th taste, which is umami, which picks up on the amino acid glutamate and gives us a meaty taste. So think about these questions: what sniffing does, where the olfactory bulb sends its information, where your taste buds are located, cranial nerves for taste, what the 5 taste sensations are, and where you can taste bitter, okay? And with that, we're going to end chapter 8.